Hi, this is Samra from ImmuneWatch and welcome to the Repertoire Room episode number 5. This episode was kind of an experiment because it was the first time that I actually recorded a live podcast at a conference, in this case the Festival of Genomics and Biodata in London. Unfortunately, what I also learned from this experiment is that I need to invest in better audio capturing devices because the quality of the recording is not what I hoped it to be. Nevertheless, I still hope that you enjoy this podcast. Welcome to the Repertoire Room, episode number 5.
I'm Samra Turajlić. I'm a clinician-scientist, and currently I'm the director of the Cancer Research UK Manchester Institute, and I lead the group currently in the Francis Crick Institute.
Okay, welcome Samra. Welcome to the Repertoire Room. We are here at the Festival of Genomics in London. Have you ever performed at a festival before?
Actually, come to think of it, when I was about 13 or 14, I did perform at a music festival.
Oh, really?
But not since then.
Playing an instrument or?
Playing an instrument, that's right.
Wow.
Playing the piano. Yeah.
That was your previous career then.
Yes.
Okay, and you just finished your talk at the Multiomics Stage. Did it feel any different than a regular talk at a conference?
Absolutely. Gosh, I mean, first of all, this place is like a jungle. There is so much happening. There's so much milling around. It's a very exciting atmosphere, I think. In the format of those talks, with everyone wearing their headphones, other than the speaker who's on stage wondering, are people really listening, is quite unusual. But, I felt that people were still engaged. I could see heads nodding. I was still getting the cues from the audience. And the fact that I knew that there was like a series of other talks going on at the same time was actually really quite exciting.
Yes. I was following your talk, and you mentioned also as well, there's a lot of emerging technologies out there. So it does kind of have this vibe of being here, connecting with all of these different types of
That's
technology.
Right. And I think the atmosphere sort of reflects the field in a way.
Okay.
Right. There's a lot of collision going on. There's a lot of newness and so on. And that's very, very exciting. Yeah.
Yes. I'm always interested in people's career paths. As you mentioned, you're also you have different positions, including a group lead at the Francis Crick Institute. But what was your career path leading into this position?
So I studied medicine at university, and when I finished and did my sort of general medical training, I really connected with oncology as a clinical specialty. And I think for me at that time, that was very much about the human experience. I felt that the place where I wanted to be as a physician was by the patient side over a long period of time, rather than kind of acute specialties where you don't really get to know people. But 2 or 3 years into my training, I sort of felt this frustration that I didn't really understand cancer biology. I kind of understood systems cancer, what was in front of me, and sort of interpreting the data I was getting from scans and so on. But I really wanted to know what was happening in the sort of cellular, molecular tissue level. So I ventured into a research lab to do a PhD. And that was kind of a deep awakening, because I would say that medicine is very, very different to science. So there was a steep learning curve for me, and often I was the laughing stock in the lab for attempting to do like 6 Western blots at the same time, because that's what medics will do. We like to multitask. But I really deeply fell in love with science, and I was very lucky. I had so many colleagues who were prepared to teach me.
Yeah.
And so I was bitten by the bug. And when I finished my PhD, I thought, well, I want to keep on doing the science, but I also miss the patients. So that's how I decided I was going to do both.
Yes, and you're still doing both, like still being a clinician and at the same time researcher?
That's right, and I think this is really critical for my program for several reasons. The first is that, for me, I think the question starts at the bedside. So the types of questions we've asked in our research program are very much about reconciling what we see, what I see in front of me in the clinic. And what I see is diversity. So, no two patients are alike. Their scans might look the same. The cancer might look the same under the microscope, the histological slide. and they will have completely divergent outcomes. And that's, that's really bewildering for the physician and for the patient and their family. So what really propelled our program at the beginning, and this is where we started using a lot of genetic sequencing, is trying to sort of deconvolve like why these outcomes are so different. And once we began to be able to reconcile that by using molecular profiling, phylogenetic reconstruction of tumors, understanding evolutionary trajectories, and therefore rationalizing these different outcomes, that became a very powerful thing for us. So that meant that we had to stay rooted in these clinical questions. But I would say that that doesn't take you away from doing fundamental science, because that's just an observation, and then you go to the lab and you figure out why.
Yeah.
So that's the reason why I continue to be in the clinic.
Yes, although I can imagine that there's like a big lag time between both of them, seeing what you see in the clinic and then trying to figure it out using basic research and science. Like it might still take quite some time to really put your finger on what was going on.
But I think this is where technologies have really helped, because in the past it would have taken a very long time to then just do very deep genotyping and phenotyping of the patient material. And now obviously this stuff is at our fingertips, whether it's tumor intrinsic or tumor extrinsic. And the other reason why I think this has to be rooted in patients to start with is that the dimensions, the temporal and spatial scales of what happens with cancer in a patient can't be recapitulated in any model. Whether it's a mouse model or in vitro model, there is always a limitation. And so we have to observe, we really have to measure what's happened in the patient. And then obviously we want to get to causality. But the observation, the ground truth is what we measure in the patient. And that's why technology is so important.
It's interesting to see this passion, and also I admire that you can combine both, because both seem very challenging to combine. But yeah, today your talk was all about multiomics platforms for especially cancer immunotherapies. Can you share a bit, I think you touched upon this already a little bit, why you think it's important to take these kind of multiomic approaches?
Yes, so maybe just to reflect on some of the points from my talk. First of all, obviously immunotherapy is a very powerful way to treat cancer. In my own practice, I've seen the outcomes for patients with melanoma transform. So I used to lose all my patients. I never got to know anyone. And nowadays, we're having discussions about, when should we apply immunotherapy and which one and so on. So it's an approach that's here to stay. But it's very imprecise, right? Because we're kind of using this systemic removal of immune checkpoints without knowing what's going to happen. So it's another free fall point for the physician and the patient and the family. Say, well, we can't tell you how you're going to do, and we don't know if you're going to develop autoimmunity or autoinflammation as a result of this. So we want to set up a program where we could do exactly what we've done before using genetics. But this time, the challenge is much greater, because the interactions between the tumor and the immune system and the host and the external factors are far more complex than the interaction, let's say, in the context of targeted therapy, where you're targeting a molecular alteration, and you can kind of mathematically very quickly deduce what's going to happen. Here, there is too much plasticity. There are so many interactors. So we really wanted to do this at scale. We wanted to do it deeply, and we wanted to use these sort of nascent technologies that are coming through to get to the depth of understanding. In the end, what the aim is to pass that knowledge to a point where we've got a set of reliable, useful biomarkers. But in this process of discovery, we really want to get to the fundamentals of what's happening in patients when they receive these therapies.
So you're using basically these approaches as maybe like a funnel. You try to scan as broad as possible, and then in hope, like, okay, we see some biomarkers that are worth it to pursue in the future and do it more targeted in the future. Then
that's right. But I wouldn't be too literal in the sort of interpretation of the biomarker just as a measurement. Obviously, it holds so valuable biological information, and let's say we're looking at something in the context of resistance to current immuno-oncology agents, that can also highlight a new target, can highlight a new mechanism, a new pathway, a new immune checkpoint, right? So it's a starting point for biomarker development, but it's also a starting point for discovery science and drug targeting.
Makes sense. I was indeed wondering, what are some of these key insights that you already gathered from all the data that you collected regarding immunotherapies? So what are some of these, the different technologies that you used, which things came up where you were kind of surprised?
Yes. So we're in the early stages yet, but I'd say we're convinced by the power of this approach and the confidence that it gives us. So one of the things that I was showing in my talk is an approach in a cohort of patients who've been successfully treated with immune checkpoint blockade. We have the tissue at the sort of post-treatment, and we want to track what's happened here. So this is where we've coupled single-cell RNA sequencing with TCR and BCR sequencing with in situ protein and transcriptome profiling, because we want to have confidence for ourselves, not for a paper, for ourselves, that this is the truth of what's happening. So here, for example, we see an emergence of an important immunophenotype, essentially, which is a memory signature. So this is a T-cell memory signature in patients who have very long-term remission from immune checkpoint blockade. So this is really highlighting this is what we need to be driving for in patients. And then when we couple that with TCR sequencing, we can also see that the clones that are emerging that carry this memory signature are new clones. They're not pre-existing.
Yeah.
And this makes sense because these patients are treated not just with PD-1, but also CTLA-4 inhibitors. And we know that the mechanism of action for CTLA-4 is that the priming is happening not in the tumor, it's happening elsewhere, for example, in the lymph node. So we can see that maintaining pre-existing clones is important with PD-1 mechanism of action, but with CTLA-4, what you're getting are some new clones as well. So you might actually be rescuing some patients that would be PD-1 resistant. The other thing that we've seen, which is with liquid, so blood-based peripheral biomarkers, is the power of high-dimensional flow. So immunophenotyping changes, across many different lineages, many different states. And here we're already seeing some hints that both at the baseline and dynamic change is separating out between patients who respond to treatment, patients who don't respond to treatment, and patients who develop immune-related adverse events. So with these are sort of the exciting nuggets we've got so far.
Of course, in your multi-omics data mix, I think you wouldn't want to pick one favorite, but we're working mostly in the TCR sequencing field, and also got to know your name because you co-authored a paper on TCR sequencing and applications in immuno-oncology. What made your lab decide to write that kind of review as well? So do you see any kind of advantages of adding this old T-cell sequencing methodology to the data mix?
Sure. So, I mean, I'd say that the TCR and BCR sequencing are sort of foundational omics in the Manifest platform, both tissue-based and periphery-based. But, our interest in TCR sequencing is more historical than that. I guess the first thing to say is that because I'm essentially a cancer geneticist, so I quite like barcoding, and that's how I viewed our original DNA sequencing studies. I was there in the early days of next generation sequencing, first time we were able to do whole genomes and exomes. And what was exciting for me, studying that in the context of tumor biology, was the ability to track what happens. It wasn't so much about, what mutations are present, but I was interested in these clonal markers that tell you the story of the tumor. Well, this happened first, and then that happened, and then this only happens on a particular background, and so on. So, I've considered that type of work kind of a barcoding exercise. And when I first came across TCR sequencing, as a non-immunologist, I have to say I had this kind of very simple view of it as another barcoding exercise. And what really inspired us to adopt it in the study that we published in 2021, the Adapter trial, was that many people in the field were saying that the T cells that you find inside kidney cancers are not tumor specific.
Okay.
That the reason why kidney cancer is full of T cells is just a very vascularized tumor. These blood vessels are very leaky, and these are just non-cancer specific T cells that are extravasating into the tumor. So we were really, when the community says something like that, you get a bit exercised. You're like, but what's the evidence for this, right? So we had a trial, which was a labor of love. It took a very long time to deliver, the Adapter trial, where we treated patients with PD-1 monotherapy. Thanks to the commitment of these patients, we were able to biopsy their tumors at the start of treatment and then again at week 9 while they're on PD-1 monotherapy. And we tracked PD-1 bound T cells and we subjected them to TCR sequencing and also single cell RNA sequencing. And this allowed us to prove that actually in patients who were responding to treatment, PD-1 blockade was acting on local T cells that were already primed by the immune system. So that told us actually at least some T cells inside the tumor are actually tumor specific. And that bodes very well for kidney cancer in terms of thinking about T cell therapies that can be extracted from kidney tumor till, but also as a biomarker. Because in patients who were going to respond to PD-1 blockade, there was increased TCR clonality at the baseline. Essentially what happened during PD-1 blockade is that those pre-expanded clones were the ones that expanded further and became cytotoxic. And we could only understand this because we were able to track the clones through TCR sequencing. Conversely, in patients that didn't respond to treatment, there was none of this clonal expansion at the beginning. And what happened on treatment is that the pre-existing clones disappeared and they were replaced by clones that were not there before, and none of those clones became cytotoxic. So this showed us the power of multimodality as well, because not only were we able to do the TCR sequencing, but we were also, through combining it with single-cell RNA-seq, we could say, well, this is the phenotype of these TCR clones. So I think this really convinced us. And of course, as a non-immunologist, I thought, well, I don't know anything about this. We joined forces with colleagues that work in that field and decided to write this review in immuno-oncology technology. So I'm really pleased that you picked that up. Yeah,
it was an easy search for me. Yes. So Samra, what's next for you? What kind of upcoming results or papers are you excited about?
Yeah, so obviously I'm excited about the Manifest program, and I think a lot will come from that. But I just want to come back to something I was saying earlier. So we'd proven that these T cells in kidney cancer are, at least some of them are tumor specific. What we don't know is what they're seeing. And the unusual thing about kidney cancer is actually it doesn't have a very high tumor mutational burden, but it is very immunogenic. It's immune infiltrated, responds well to immunotherapy. So we're now exploring all sorts of classes of mutations and potential antigens, including the dark genome. And in the process of doing that, we want to deorphanize some TCRs that we think are important. Now, how do we know that they're important? These are TCRs that are expanding under successful immune checkpoint blockade in patients. And again, due to patient participation, we're able to sequence these TCRs. Again, think about what kind of signatures we're looking for, tumor reactive signatures. And then we will screen them against around 2,000 different antigens per patient. And of course, once we get to that ground truth, we're able to use that kind of data to iterate models of antigen prediction as well. And we really want to answer this question about what's making kidney cancer immunogenic. So watch this space.
I'm super excited to hear more about it. So I'll definitely keep an eye on what you'll be producing, maybe next year at the Festival of Genomics again, with an update on all of that. So where can people find out more about you and your research if they really want to learn more?
Yes, so do please get in touch if you think that this is of interest in terms of collaboration. Otherwise, do have a look at our lab webpage at the Crick Institute, at the COK Manchester Institute, and then finally the Manifest Consortium has its website as well.
Okay. My final question is always like, you have built this career in science, but what would you have done if you were not working as a scientist nowadays?
Oh gosh, that's a very difficult question. So I really love the arts. So I imagine thinking back to that music festival, maybe something in the arts.
Okay.
Yeah.
Thank you, Samra. I really enjoyed this conversation, and I hope to see you in the future as well.
Thanks very much for hosting me.